We're back. This is part 2 of Enzymes, the STEM case study, where a dog is having trouble with its digestion. In our first part, we looked at enzyme reaction rates, how enzymes speed up reaction rates, and how they catalyze reactions by making them go faster. Let's look at enzyme structure now so we understand how enzymes function. There are 75,000 different enzymes in the body, and each one has a specific job. Without even one enzyme, if you have a genetic disorder, you can have serious health consequences. Enzymes are a type of protein. Not all proteins are enzymes, but all enzymes are proteins. Like other proteins, they're made of small building blocks called amino acids. I think about amino acids as beads on kind of pop beads. The amino acids pop together. They all have different sizes and shapes, and they stick together with bonds. Each enzyme has a different sequence of amino acids, so every enzyme has a different structure or shape. So we can see the different enzymes and how they have different amino acids, which are different sizes and shapes and have different charges. So they result when the protein folds into different shaped enzymes. And if that enzyme doesn't have the correct shape, it won't work. We say in my class, lose your shape, lose your job if you're an enzyme. Every enzyme has an active site. That's where the substrate or the thing it's working on fits. So here are the active sites in these enzymes. It's highlighted in yellow here. Each enzyme's active site has a different shape. You can think of this as being the lock. Just like you need a specific key to fit into a lock, only reactants with a specific shape can fit into an enzyme's active site. Reactants that bind or attach to an enzyme are also called substrates. So substrates are what enzymes work on. You can see that those substrates, or what the enzyme's working on, fit like a key into a lock. They fit very tightly. Which of these molecules will fit this enzyme? Well, we can drag a few over and try. Well, that's probably not going to work. Incorrect, it says. Try again. Let's try a rounder shape here. That's not going to work. Doesn't fit very well. Let's try this shape here. Yep, that's the one. Once the substrates are bound, the enzyme catalyzes the reaction and converts the substrates to products. Notice in some cases that the enzyme is tearing things apart or pulling molecules apart, and sometimes it's putting molecules together. Which of the following is false about the structure of an enzyme? Every enzyme has an active site. Well, that looks pretty true. An enzyme can catalyze most reactions if the reactants are at high concentration. Well, we haven't heard anything about concentration yet. An enzyme's active site is determined by the shape of the enzyme. Enzymes speed up reaction rates. An enzyme's active site is determined by the shape of the substrate. It has to fit the substrate that it's working on. So this one seems the most false. We haven't heard anything about concentration rates, so let's pick this one. All answers are true except B. An enzyme shape only allows certain substrates to fit into the active site, so it will only catalyze certain reactions. If the substrates do not fit the active site, no reaction will happen, even if they're present at a high concentration. The shape of an enzyme also depends on temperature and pH. Even slight temperature and pH changes can cause the change in shape of an enzyme. This shape change can stop the enzyme from working properly. Remember, lose your shape, lose your job if you're an enzyme. Enzymes are kind of like rock stars or opera singers or celebrities. They won't work unless things are just right. It has to be just the right temperature and just the right pH. pH is how acidic or basic something is. So if we increase the temperature, the enzyme changes shape and it won't work. The active site changes shape as well, and our substrate doesn't fit. 37 degrees is body temperature for humans, and that's all human enzymes should be at 37 degrees. They work best usually at that temperature. If we decrease the temperature, the substrate doesn't fit the active site. This is why hypothermia, which is being too cold in this case, or hyperthermia, being too hot, is really detrimental to your health. Your enzymes can change shape and not work anymore. If things are at the wrong pH, the enzyme can change shape as well and not work. Your body works hard, and dogs do this as well, to keep your blood around the same pH so that your enzymes have a stable environment in which to work and they continue to do their jobs. If your blood pH gets too off, your enzymes can stop working. So the takeaway here is that enzymes need a particular temperature and a particular pH to work. They're very specific about the conditions that they will work in. So they're kind of like fussy rock stars or celebrities. They will only work if it's just right. The correct pH and the correct temperature. So now that you've learned about enzymes and metabolism, return to the case to investigate Claire's metabolism problem. Let's go to the case and let's try to help Claire. Claire's eating a lot of food, but she's losing weight. This suggests that food is not being broken down by Claire's enzymes. The process of breaking down food by enzymes is called digestion. There's Claire's stomach. There are some enzymes. Food contains many nutrients, including proteins, carbohydrates, and lipids, aka fats. Well, part of lipids are fats. All of these things can be found on a nutrition label. The lipids will be found under fats on a nutrition label if you check a nutrition label on the, your package of noodles or rice or cookies or whatever you're eating. During digestion, enzymes break down these nutrients into small molecules that the body can absorb or take in. The nutrients have to cross into your bloodstream across a small layer of cells in your digestive system. So you can't have big chunks of food going across cells. You need to break them down into individual molecules and molecules that aren't too big. So let's feed Claire and see what's up with Claire's digestive system. In the stomach, proteins in food are broken down by an enzyme called pepsin. It's the same for human stomachs. If you've ever puked, you know that your stomach is very acidic, and pepsin only works at a very low pH, a very acidic pH. Pepsin is released by cells in the lining of the stomach and works best at a low acidic pH. Here's pepsin. The stomach maintains that low pH that pepsin needs by releasing hydrochloric acid. So once it leaves your stomach, food goes to the small intestine in Claire and in people. See what happens to the food in the small intestine. Enzymes in the small intestine break down proteins, carbohydrates, and lipids. These enzymes are named after the substrate they break down. Proteases break down proteins. Carbohydrates break down carbohydrates. And lipases break down lipids. You can tell what the what substrate the enzyme is working on by the beginning of the name here. Pro for proteins, carbo for carbohydrates, and lip for lipids. So in the stomach, we've got pepsin. It requires a low pH. It primarily works on protein substrates. In the small intestine, we have proteases that break down proteins, lipases that break down lipids, and carbohydrates that break down carbohydrates. Let's collect some data from a healthy dog. To do this, we'll take 10 milliliters of liquid from a healthy dog's stomach and 10 milliliters from its small intestine. We'll now add 1 milliliter of the liquid from the stomach to a solution of 1 milliliter of protein, the substrate. So we're going to digest some protein with the stomach fluid. So this graph shows how the protein concentration changes over time. When 1 milliliter of stomach liquid is added to 1 milligram per milliliter of protein, the substrate, what the enzyme is working on, what the enzymes in the stomach are working on. As the enzyme breaks down the substrate concentration goes down. So as the reaction goes on, as the enzyme catalyzes the reaction, there's less and less protein in the stomach. So the amount of protein is going down. The data from the healthy dog is your control data. So we're comparing Claire to a healthy dog. We're going to run the same experiment on Claire. It looks like Claire's stomach proteins are okay. Stomach acids, stomach enzymes are okay because she's got the same data as a healthy dog. Compared to a healthy dog, did the enzymes from Claire's stomach break down the protein at a low, normal, or high rate? Well, since she's at the same rate as a healthy dog, let's say normal. Now we need to examine the enzyme activity in the small intestine. This is a little bit more complicated because remember there are 3 enzymes we're looking at in the small intestine: carbohydrates, protease, and lipase. 1 milliliter of liquid will be added to solutions with either protein, lipid, or carbohydrate. Remember that the enzyme breaks down the substrate, so the substrate concentration goes down. You can see the healthy dog, the amount of proteins, lipids, and carbohydrates are going down pretty quickly. Did the enzymes from Claire's small intestine break down the protein at a low, normal, or high rate? So here's Claire in red. Claire's the red line, and the healthy dog is the yellow line. The healthy dog is going down much faster, so the healthy dog is digesting more quickly here because the amount of protein in the stomach is going down pretty quickly as the small intestine proteases, the protein acting enzymes, break down the proteins in the dog's small intestine. The same is true for the lipids and carbohydrates. So Claire has got a low rate of digesting all of those molecules because her line doesn't go down as fast. Claire's got kind of a smaller slope. Remember, the slope shows the rate. The slope is how level the line is. The more tilted the line is, the greater the slope. The healthy dog has a higher slope, and Claire's is kind of more gentle, which means her rate is lower than the healthy dog. Where is the problem in Claire's digestion? Well, we see the healthy dog and Claire right parallel here, but we see the healthy dog and Claire are not together over here in the small intestine data. So we seem to have a problem in the small intestine. So there could be 3 problems Claire is having. She could have active site mutation, abnormal pH, or low enzyme production. And in part 3, we'll investigate all of these problems.